Moving on to the semilunar valves, the aortic and pulmonary valves. The single outward tract, or the truncus arteriosus, divides to form the aorta and the pulmonary trunk. In each divides a valve with 3 cusps. Importantly, these cups are named based on their embryological origin and not necessarily their final adult anatomical position. So we have the pulmonary valve that has the right, left, and anterior cusps. We have the aortic valve, which has also the right, left, and posterior cusps. These are clinically critical because the space behind these cups, or the aortic sinus, are the origin points for the coronary arteries. The right aortic sinus gives rise to the RCA, and the left aortic sinus gives rise to LCA. And the posterior aortic sinus gives rise to no coronary artery, hence it's called the no coronary sinus. On the right, we have a transverse CD angiogram that shows the view of these origins of where they come from. So the RCA over here, LCA over here, and the diagram on top shows you the positioning relationship of these valves, whether they're anterior, posterior, left, and right. The cardiac skeleton is also known as the cardiac fibroskeleton. This is a dense network of fibrous connective tissue that's situated crucially between the atria and the ventricles. It serves to 2 vital functions. First, it provides structural support, maintaining the shape and patency, the openness of the valve's orifice, and serving as attachment site for the valve's cusps and the heart muscle itself. Second, it also plays a role as an electrical insulator. It prevents electrical impulses from the atria directly passing into the ventricles, except through the designated AV bundle pathway. The skeleton is composed of 4 fibrous rings, or what we call the annuli fibrosi. Each surround the 4 heart valves, the right AV tricuspid, the left AV mitral, the aortic semilunar, and then the pulmonary semilunar. These rings are interconnected by particularly 2 main fibrous trigons. We have the right fibrous trigon. This is located between the aortic and the right AV rings, and the left fibrous trigon located between the aortic and the left AV rings. These trigons serve as an anchor point, significantly contributing to that mechanical stability of the heart. On the right, you can see how the atrioventricular bundle goes through right in the middle, and then we have the right fibrous trigon right there, and we also have the left fibrous trigon right there, the smooth groove where it attaches to. And this is important for mechanical attachment. And you can see also the rings as well. The right atrioventricular ring, a left atrioventricular ring, and then we have the semilunar pulmonary ring, and then the aortic valve ring. Let's revisit the general structures of the heart valves. Established, they are fundamentally composed of 3 distinct layers of connective tissue. They all covered by the endocardium, the heart's inner lining. These valves are firmly anchored to the fibrous rings of this cardiac skeleton. You can see on the right that the valves are attached to the fibrous skeleton over here. Their attachment right there, they're attached. And it shows that the deep within the endocardium is in the subendothelial layer lie the Purkinje cells. So over here, the Purkinje fibers lie within it, and this is at the terminal part of the conduction system. The core 3 layers of the valve leaflet are going to be the fibrosa, the spongiosa, and depending on the valve surface, either the ventricularis, which is on the ventricular side of the semilunar valves, or the atrialis on the atrial side of the AV valves. Over here, the DCT stands for the dense connective tissue, or the layer of the subendothelial cells, and then the N region represents the innermost squamous cell layer. These are the sublayers of the mitral valve. So a deeper dive into these key layers. Let's start with the fibrosa. So think of this as a strength layer. It's located on the ventricular surface of the AV valve, the tricuspid and the mitral, and on the atrial surface of the semilunar, the aortic and pulmonary. Its primary function is to provide tensile stiffness to the leaflet, and it's derived from the fibrous rings. It's composed mainly of dense type 1 and type 3 collagen. Critically, it's in the AV valves, and the fibrosal layer extends directly into that extension of the heartstring called the chordae tendineae, and these fibrous cords are shown in that gross image on the right. They then connect to that bed of papillary muscles within the ventricles. You get the pectinate muscles in the atria as well as you have the papillary muscles in the ventricles. Next layer is called the spongiosa. This is the middle shock absorbing layer. This layer is made out of mostly loosely arranged collagen elastic fibers, as well as a lot of ground substance and proteoglycans. The looseness allows it to help dampen any vibration during valve closure and generates flexibility. As seen in the histological image of the mitral valve, the spongiosa layer on the right side, as you can see, is really, really thick towards the free edge of the leaflet at the end, and that helps to properly prevent any backflow of blood. So it plays a role for preventing any regurgitation. So you can see over here the different layers. On the atrial side, we have the atrialis, spongiosa, and then fibrosa on the ventricular side. Next, we have the ventricularis or the atrialis layer, depending on which chamber surface it faces. We call this layer the flux layer. It lies adjacent to the ventricular or atrial surface and is covered by the endothelium. It's composed mostly of connective tissue that's dense, rich in elastic fibers, and its main function is to facilitate the valve's movement by allowing the leaflet to extend and recoil smoothly during that cardiac cycle. One special feature, particularly in the AV valves, is the presence of some of these smaller cardiac muscle cells within this layer. And they're actually derived from the atrius, not the ventricles, which help modulate that leaflet stiffness. One of the key characteristics that apply to all the valves leaflets is that they're just avascular, meaning that they don't have any blood supply, and they instead allow diffusion from the blood to go directly through them. The cells residing within these valve leaflets are unique. These are what we call the valvular interstitial cells, the VICs or VICs, and these cells maintain the valvular structure and health throughout their life. They originate from the endothelial cells, but they resemble more of a fibroblast microscopically, so that kind of collagen spilling cell. And the VICs express a protein called the chondromodulin 1. This protein actually inhibits blood vessel formation, so it keeps it as an avascular state. Over here, just another view of the aortic side on top, and then the ventricular side on the bottom. We have the fibrosa layer, spongiosa, ventricularis, and then within that layer you have all these interstitial cells. These are going to be, you're going to find interstitial cells in here, as well as a layer of smooth muscle cells, and then they resemble more the fibroblast cells. Now let's focus on the semilunar valves, the pulmonary and the aortic valves. Each of these valves has three cusps, and they appear concave when viewed from the great artery, looking back towards the ventricles. And as we already named them before, the pulmonary has the anterior, the right and the left. The aortic has the posterior, right and the left. And a major difference from the AV valves is that these cups do not have those tendinous cords for support. Their mechanics are relatively simple, as shown in the diagram on the right. During ventricular contraction or systole, the high pressure pushes that cusp open, allowing the blood to eject into the aorta or the pulmonary trunk. And then as the ventricles relax, the pressure drops down, and that brief backflow of blood from that elastic recoil allows the great arteries to catch the pocket-like cusp, causing it to snap shut. This then prevents any blood from flowing back into the ventricles. So over here on the right, you can see when it goes out, opens up, and then as it closes, slaps shut, it pulls downwards, and then the blood is able to fill up. Let's look closer at the cusp anatomy. The free edge is thickened to help ensure that there's a tight seal when it's closed. This crescent-shaped thickened red edge where the cusp meet is called the lulunae. And at the very center of the lulunae is a small, little thicker nodule. Behind each cusp, there's going to be an origin of the aorta and the pulmonary trunk. And these are dilated spaces called the aortic and pulmonary sinuses. These spaces then form between the dilated artery wall and the cusp itself. So functionally, they create these small little openings that prevent any valve cusp from being plastered against the vessel when it's open. This ensures that they can also close quickly. Clinically, the aortic sinuses are really important. Remember, the right aortic sinus gives rise to the right coronary artery, and the left aortic sinus gives rise to the left coronary artery. Whereas the posterior aortic sinus does not have any coronary artery origin. And this is what we call the non-coronary sinus. Over here on the right, you can see a view of the different leaflets, the pulmonary valve, aortic valve, and the different leaflets. So the right coronary leaflet, left coronary leaflet, and then the non-coronary leaflet. The tricuspid valve over here, the superior leaflet, the septal leaflet, and the inferior leaflet. And then the mitral valve, the anterior leaflet, posterior leaflet. Now let's briefly look at some of the CT scans. So these images are derived from CT angiography. In letter image A, this gives an overview showing you the RCA giving off several branches, the conus branch, which oftentimes supplies the top part of the right ventricle's outflow tract, the RV marginal, and also the acute marginal branches, which run along the right ventricle's free wall. In letter B, you can see very clearly in a CT scan the three-dimensional view that highlights the RV marginal branch supplying that free wall. And letter C demonstrates anatomical variation. So remember we said that there's a lot of variations in populations, and it's a very common feature in coronary circulation. This particular patient has a conus branch, but this displays actually three distinct RV marginal branches. So there's three subbranches of 1, 2, and 3 coming out of the RCA. Let's take a look at another one. So this slide illustrates a coronary dominance using a CT angiogram. Dominance, again, is defined by whichever artery gives rise to that PDA, the posterior descending artery, which runs on the posterior interventricular groove and oftentimes then supplies the posterior lateral branches to the left ventricle. In image A, you can see the right dominant system, where there's 67% of the people. The RCA continues around the back of the heart and supplies both the PDA and the posterior lateral branches. That's also known as a PLLV. And then in letter B, you can see this is left dominant, about 15% of the population. In this case, the circumflex, the CX artery, is a branch off of the LCA, and it wraps around and gives rise to the PDA. So it wraps around here and gives rise to the PDA. And then in letter C over here, this is codominance, about 18% of the population, and that RCA supplies the PDA. RCA comes down to the PDA, while the circumflex artery wraps around and supplies that posterior lateral branches, the PLLV. And these slides here present a collection of images that you should review for the practical. They could be used for an exam question. They have different views of the veins, as well as some of the key chambers of the heart, as well as the arteries. These are from your Theme 1 image bank. And last but not least, a quick brief overview of the cardiac cycle. There are two main periods for the ventricle. There's a systole, where the period is when the ventricle contraction and emptying occurs, and diastole, where the period of ventricular relaxation and filling occurs. The table below kind of highlights some of the key events in the cardiac cycle, which you'll take a deeper dive from letters A to G, and they correspond to another diagram called the Wigner's diagram that's oftentimes used to display how it changes with pressure. This is not going to be shown here, but it starts off with letter A, which is atrial systole. This is the final phase of the ventricular filling, and it progresses through the ventricular contraction. That then leads into the letter B, the isovolumetric contraction. This is going to be a phase where the ventricles contract and the ventricular pressure increases, and also the ventricular volume is constant, leading to all the valves being closed. Brief moment there. Then letter C is called the rapid ventricular ejection. During this phase, the ventricles contract, ventricular pressure increases, gets to maximum, and the blood gets pushed out. The aortic pressure also increases and also reaches maximum as well. Then following that leads into letter D. This is going to be the reduced ventricular ejection. The ventricle's blood have been already ejected at a slower rate at this point, and the volume has reached a minimum. The aortic pressure begins to drop down and runs off into the smaller arteries. Then that leads into the isovolumetric relaxation, which is letter E. This is another brief moment when all the heart valves are closed and the ventricles are relaxed, the ventricular pressure decreases, and then the ventricular volume is relatively stable. So both isovolumetric moments in the cardiac cycle are very brief. Then that leads into letter F, rapid ventricular filling, where the ventricles are relaxed, the ventricles fill passively from the blood from the atrias, and now the ventricular volume slowly increases, but the pressure is still relatively low and constant. Then that leads into the final phase, letter G, which is the reduced ventricular filling, also known as diastasis, where the ventricles are relaxed and it shortly resets again. All right.